Broadcast is now starting. All attendees are in listen-only mode.
Good morning, good afternoon, and good evening, everybody. My name is John DeBella, and on behalf of Simulations Plus, I welcome you to today's webinar, where we will describe and demonstrate the exciting new features in PKPlus version 2 and show how it provides all critical functions necessary to be your in-house PK modeling platform. I will present an overview first of PKPlus using a few slides, and then hand over controls to the PKPlus product manager, Mark Flieger, who will demonstrate the platform live. Joining Mark and myself as panelists for the Q&A session at the end are Viera Lukachova, Director of Simulation Sciences, and Kay Zato, Senior Scientist 2 in the Simulation Technologies Group. An opportunity to ask questions will take place at the end of the webinar. You may either type your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. This webinar is being recorded for playback at the online resource center on our website, www.simulations-plus.com. Also, upon request, the presentation slides can be made available and shared with colleagues. With that, let's get going. And before I dive into the slides, what I'd like to do is a short demonstration of one of the new features in version 2, which is running now a series of command line scripts to verify the results on your system are matching with the results that we would expect upon installation of PKPlus. So we've now included with every installation of PKPlus a validation toolkit. After you install PKPlus, there will be a verification subfolder available. Double-clicking and opening that folder, you'll see that there will be a series of Windows batch files, several of them based upon whatever system locale you're using on your machine. In my case, I'm running off of Windows English systems locale, so if I double-click on this batch file, it will then open up a command line prompt and ask me for my user password. One of the other new things with version 2 of PKPlus is we've introduced Windows authentication when logging into PKPlus. So if I type in my Windows password, the program now will automatically go through a series of 25 analyses for a variety of different applications, whether it's single dose, steady state, non-compartmental analyses, compartmental fits. And as it's going through and setting up the projects and doing the mapping and generating the results, it will compare those results with the results we would expect based upon all of our internal runs and calculations. And we'll see here as I come back to this in a couple of minutes, we'll get a full report of these comparisons and see whether or not we have any issues. So as that's running, let me transition back here to the slides and go through a little bit of background here with PKPlus. And with PKPlus, what we like to say here is that this is a true plug-and-play PK modeling platform. It really is going to provide you with all of the necessary functionality that's required for doing your NCA, your compartmental PK fitting. And now in version 2, we've extended that to allow you to do simulation or prediction activities as well. So let's start a little bit first with the what and the why of PKPlus. PKPlus is a platform that's going to combine sophistication. And by sophistication, we just mean providing you with the key elements that are going to be required for doing all of your PK analyses. Powerful non-compartmental analyses options, efficient compartmental analysis routines. And now in version 2, as I mentioned earlier, the ability to do predictions. We've incorporated both non-parametric superposition options and also compartmental multi-dose simulations. So once you've done your fits with some existing PK data sets, we can do a lot of what-if hypothesis simulation activities as well, just by clicking in a few locations within the interface. And also under this idea of sophistication, we've strengthened the PKPlus platform so that it can assist with your internal 21 CFR Part 11 compliance systems. In version 1 of PKPlus, we already provided you with audit trails and validation aspects. Now what we've done is extended that to incorporate authentication through Windows operating system logins and also group settings. So now your PKPlus administrator can provide access privileges at different levels to different users. And as you saw starting off this webinar, we've also incorporated this automated system verification routine where we've provided over nearly 25 different data sets and set up command line scripts so that in a matter of minutes, you're able to verify that your system has a correct PKPlus installation in place. So that's the sophistication aspect. But as we released version 1 about a year ago, and as we received feedback from the many companies that have been using PKPlus during this 1-year period, we identified ways in which we could make it simpler. We don't need to have all of the bells and whistles and options that other platforms might have. When we're really focused on just providing these PK applications and routines, we can simplify things. And so we've already had in place point and click interface routines to minimize user errors. We've already had in place the ability to import common data files very easily. We've gotten feedback from users over the past year about how much they appreciate the exploratory data analyses features and how straightforward it is to create analysis data sets. We have made some improvements, which we'll talk about in terms of creating report quality tables, figures, and lists. And one of the big things that people wanted us to try and incorporate in version 2 was the ability to save and reuse custom templates for mapping, for reporting, so that users could just go in with those templates and for analyses that would be coming in, reuse those to quickly generate their results. And we've also tied that ability to create and save those custom templates with command line script options. So now there's even opportunities for you to do these PK analyses without going into the actual PKPlus user interface. So we really feel like we've combined the sophisticated aspects that are required for a PK modeling platform, but simplified it in a way that just gives you exactly what you need. Now, how does this benefit you? Well, from an industry perspective, if we're talking about folks who are in the deep DMPK environment or preclinical PK study managers who are maybe performing high throughput animal studies, you guys need to generate quick and accurate PK study reports in either a validated or non-validated environment. And maybe what you'd like to do is also be able to simulate or predict in animals what would be the exposure at different doses for different dosing regimens. And so we've incorporated this ability to do the simulation routines as well right within PKPlus. For the clinical pharmacologists who maybe are analyzing large amounts of data in a validated environment and trying to communicate these results to non-modelers or other members of their team, we can manage who can do what with the data sets. As mentioned earlier, there's now tools for PKPlus administrators to give different access privileges to different user groups. And also we extended the ability to customize the variable and plot settings and reuse those so that you can create the or present the results in such a way that makes it easier for you to communicate this information over to other team members. And for the modelers who might need to generate or produce parameter estimates for GastroPlus PBPK models or non-mem population PK models, it's very easy to set up analysis data sets And as we've done in the PKPlus module of GastroPlus, you can automate the fitting of different compartmental PK models and use those model estimates as inputs into some other routines. So there's a lot of benefits here for folks at different stages of the research and development process, and for industry. From a licensing perspective, every PKPlus user license is treated as a floating seat. No ifs, ands, or buts about it. You don't have to specify to us who would be part of a group that might be sharing a license. IT installs the software, and then anybody at your site who has interest in using it does a local installation and connects to that network server environment. So this can be shared across or within departments. There's no optional modules. The verification toolkit that we have now with PKPlus version 2 is part of the base installation. So you're able to validate your systems and the results that are being generated without having to license any additional suites or toolkits. And as was the case with version 1, user license costs are a fraction of competing tools. Now, from a nonprofit or academic perspective, for students learning pharmacokinetics, or professors teaching pharmacokinetics, or researchers on government or nonprofit grants who need to perform basic but thorough PK modeling activities as part of their projects, we're very happy to announce that starting with PKPlus version 2, there is a new no-cost academic program. All you have to do is apply. And you can see the URL here on our website. So now you're able to access the same functionality and features from PKPlus version 1 with the additional enhancements in version 2 at no charge for academic nonprofit use. So this is really exciting. Lots of key enhancements that we're going to talk about now coming up, and all of this at a very low cost for industry use and for free for academics. So now let's talk a little bit about what's new in PKPlus version 2 specifically. This is a list of the highlights. We've incorporated new and improved application level compliance features. As I just have demonstrated, there is the command line scripting with over 20 PK data sets for the system verification. And we've also switched now to Windows authentication and set up user group options for PKPlus administrators. We've added non-parametric superposition and compartmental multi-dose simulation capabilities. And for the compartmental multi-dose simulation capabilities, we've actually mirrored some of what you can do in GastroPlus when working with mixed multiple dose files, where you can simulate here combinations of intravenous and extravascular or oral administration at different dose times, at different dose levels. You have the ability to save custom mapping and report setting templates for use in other projects. And even more exciting is that these can be called from the command line scripts now that are available with PKPlus version 2 to automate the processing of your source files. So imagine a scenario where you're in a high throughput environment, maybe doing rat studies for a library of 20 drugs. You can take those source files, either for each drug compound, run them through a command line script to generate your NCA report without having to worry about working through the GUI. That's really powerful. And it's something that we hope to extend to some of our other applications coming up over the next couple of years. There's improved data handling from source files during import, the ability to create custom variables to store data, improved plotting flexibility, and of course bug fixes. Now, diving in here in a little bit more detail on a couple of these items. The first one is with respect to having an application that can fit within a 21 CFR Part 11 compliance system. This is something that Walt Woltos, the CEO of our company, has tried to emphasize over the course of the last couple of years as we've been working with PKPlus as a standalone tool. And that is to remind everybody that it's not possible for any vendor to offer a turnkey Part 11 compliance system. At best, what we can do is offer an application that is going to contain the required technical elements of a compliance system. And so with PKPlus, we have all of our internal documentation and SOPs that describe the software development process and testing. And then we can also provide, as part of the system, audit trails where we have date and time-stamped user logs with all major actions tracked. We can archive results in locked files that can be viewed only by those folks which have certain access privileges. We've done validation of the platform, doing extensive testing through a variety of options against other well-established software methods. So internally, we have a much larger data set. We've scripted equations in R, in Excel, and used other methods to calculate results and compare those results with what we're generating in PKPlus to validate that the equations in the output from PKPlus are accurate and correct. We've introduced now, as part of version 2, an authentication and group settings feature with the ability by default now to use Windows authentication logins, and then, as mentioned earlier, allow PKPlus administrators to manage project privileges. And then we'll see here in a minute this automated system verification, where now we have our tutorial set of 25 data sets where we can run these through command line scripts to confirm that your PKPlus installation produces the expected results, that your results are matching up with what we have generated. So now we feel like we have hit on all of the major items, all of those major technical elements that would make PKPlus an application that could be part of a compliance system. Now, with respect to those command line scripts for either doing verification of your installation or automating some of the routines that you would like to run through the program, these scripts can be set up using open source XML editors like we see here on the left, or just translated into simple command prompts using a DOS interface. And the objective with using the command line scripts would be to verify new systems with our scripts upon installation, or automate the PK analyses with your own custom templates. Again, imagine a scenario where you've got a large set of PK data, whether it's for different drugs during a quick early screen or in a clinical setting, and we want to be able to just load all of this data in and produce some set of reports, whether it's non-compartmental, compartmental fits, or even simulations themselves. And we can do all of this within the context of our command line scripts. For the non-parametric superposition, we've introduced both single and multiple dose options, and Mark is going to demonstrate this a little bit here in a couple minutes live. But in the PKPlus interface, you'll see on the analysis tab, when we select non-compartmental analysis, there will be this new area here for working with a non-parametric superposition method. And as mentioned, what we can do is work with both single and multiple dose options. So you're going to have a variety of different interpolation methods that you can select from. You're able to define the half-lives to reach steady state, set up loading and or maintenance doses, define your dosing interval and number of doses, and then produce either a single dose output or multiple dose output. And then with that output, we can also easily calculate the non-compartmental statistics on these profiles. So we can use these predicted curves to also generate what the NCA information would be theoretically. From a compartmental PK simulation perspective, same analysis tab, but this time selecting the compartmental subtab here. We've had the ability to do the fits for different compartmental model types using linear or nonlinear approaches. And now what we can do is set up a dosing regimen. And as mentioned earlier, this can be mimicked, or what we've done is tried to mimic some of the capabilities from the mixed multiple dose support file from GastroPlus here, where you can use a combination of different dosing routes, dose amounts, start times, intervals, and infusion times. And then based upon our values here, whether they are from a compartmental fit or some initial values that were generated through some other method, we would be able to then simulate in a compartmental manner what the PK curves would look like. And just like we could do with the non-parametric superpositioning, we can generate the full statistics following these compartmental PK simulations. So these are just some of the new features, the highlights here in version 2. Before I transition over to Mark and let him demonstrate the platform live, let me just go back for a second to the verification scripts that are now part of PKPlus version 2. So we've gone through and run 25 different analyses here. The prompt is saying to press any key to continue. And in that verification subfolder, there's going to be a results subfolder. And here in that folder is a compare subfolder, which will have a timestamp associated with it. And when I double click, it will then show me all of the output files for the various analyses that were run. Single dose, steady state, non-compartmental, compartmental, bolus, tablets, different methods, linear, linear log, linear up, linear log down. And if I double click on any of these output files here from this comparison verification run, what I'm hoping to see is that they open empty. In that case, that would then be the indication that the program found no issues for this particular run, that the results generated from this run on my system match up exactly with what was expected from the PKPlus results. And so I could navigate through each of these files and hopefully see, when I open them up, a blank window here, indicating that the results are matching up well. If I do see some text in here that would indicate that there was a mismatch between the results generated, we can then provide some assistance and some guidance trying to explain and understand what went wrong with the installation. So with that, let me pass the controls over to Mark and ask him to take us through a demonstration live of some of these new features and functionality in the PKPlus platform. Mark?
All right, thank you, John. I'm going to go through a product demo and kind of start to finish. You'll see some of the existing features that were in the previous version, but we'll highlight some of the new features. So here's the screen is showing where the Windows authentication has been added. So I'm just going to add my password and log in. And I'm going to create a new project, and we'll just call this webinar. And if you want, you can add a description, and we'll just put something in simple for webinar. And PKPlus will open a blank project with the Files tab enabled. So the first thing we want to do is load a source file. So on my desktop, I've got a source file here. And this source file has some data we don't want in it. And this is going to show one of the new features. The message comes up saying we can load the file, but it's not suitable to use. And you'll see we have a little comment data here. PKPlus assumes the first row is a header row, but in the case of this file, we have some extra data. So what I'm going to do is actually change my header row and exclude out the data I don't want and just finish the import. And that lets us get rid of any data you may not want, just some simple editing to make it easier for you. But once we have our data imported, what we need to do is map these columns to our source variables. And so you'll see simply selecting the source columns to line them up with our variables. And the new feature that was mentioned was the ability to save this mapping for reuse. So what we'll do is we'll load a saved map. And once we do that, you'll see that all of the mapping is complete, as well as any unit columns we have mapped. And if you were to modify this, you could save it under a new name and use it later if you had some small changes between your file formats. By clicking Apply Map, we'll notice that it found a variable it's not sure what to do with. In this case, IV. It can't determine whether it's infusion or bolus, so we're going to tell it's bolus in this file. And then it'll complete the application of the map and sort our data. From here, we can take a quick view of how the data mapped, and it gives us a little notes that we have a couple columns that have some non-numeric data. In this case, we have some blank spots and some other data that's missing. And we can handle that later on in the program. So what we're going to do now is once we click Save Data to Database, it's going to go through and parse our data and break it up into our individual profiles. And it'll move us to the preview pane. And this is where we can get into our exploratory data analysis. By default, it opens to subjects, and we can look and see the individual profiles that match to a subject. But what we can also do is use the automated groups feature. When PKPlus analyzes the data using your mapping, it determines some groups automatically. So you'll see ones based by dose level, by the route. So you can make your own groups or use the existing groups. What we're going to do is open this infusion, and you'll see we have some missing data in these fields. And so what we want to do is handle those. You can go through individually and remove them, or you can set up a BLQ. In this case, we know the value is missing, and we'll treat it as missing for the full profile. What it'll do is it'll go through this group and apply that BLQ. So now we'll see it's removed those points from the data we're going to use. We still have a record of it, and it shows that it's been BLQ'd, but we won't need to use it in our analysis. What we can do is if we like this data, we can make it an analysis data set. So we'll just call this infusion. And that'll move it over to our analysis tab as a data set. But if you want to create your own group, you just click the Add Group button, and we're going to create a fasted 140 tablet group. When we create this group by default, you'll see that it contains all of the profiles and all the subjects. But what we can do is filter this down, and as I do this, pay attention to the profiles and you'll see what happens. So we're going to pick our fed fasted variable we mapped, and we're going to say we want all of the fasted ones. And then the dose level we'll pick is 140. And then the route we will select tablet. And you'll see we're down to 25 profiles and 25 subjects. And I clicked an extra one and I can take that out. Now if we look here, you'll see our group is narrowed down to exactly what we asked for. And we've added some improved plotting functionality. So you can actually hover now and it'll highlight which profile you're looking at, but also update the pane with details on that profile. Anytime we're in this plotting engine, we get our live data as well as statistics and an updated plot. This will update with our selection. So you can select individual profiles and get details, statistics, and plots only for your selection. A simple Control A will highlight it all again. We've also added a feature where you can manually select your data. So in the case of a data set, If you wanted to see the distribution by age, you could simply select age, count, and it'll give you the count per age. You can save this image as a bitmap, JPEG. You can also save the data, copy it to the clipboard, and it saves it as tab delimited data to create your own reports or notes as you go. So we're going to use this group as well as a data set. So we're going to copy it over to the analysis data set. And for those who aren't familiar with the program, groups and data sets have a slight difference in that groups can be modified and they're used to explore your data, where data sets are analysis data sets. You can do much of the same thing you do with groups, explore your data, but once the data set has been used in analysis, to maintain the audit trail, it can no longer be modified. So we're going to start with our FASTD 140 tablet data set, and we're going to set the terminal slopes because we need terminal slopes to do some of our operations. We set the initial method, which will be the auto method, and it'll go through and calculate our terminal slopes, and we can view each one independently. We can add or remove points to see if our results improve, and you can do that in the table or the plot. Once we have our terminal slopes, in this case we're going to add our partial AUC range as well. So we're going to go 0 to 12 hours, and we'll do 12 to 24. And once we have our settings that we like, we hit Run NCA. The NCA runs, and our results appear. You'll see we have our partial AUCs in our table, as well as individually for each profile we select. And much of the same functionality is there. You can save any of your views and any of your data to use in your own reports or in your notes. Once we've run an analysis, we have a results over in the results tab. So by selecting this result, we'll get a top level view of the settings we used. We have the ability to add notes for ourselves, and we can also generate some auto reports. So what I'm going to do is I'm going to select some of our auto reports, check the box that'll open the folder where I put them, and I'm going to put them in just the results folder I have set up. You'll see it's generating the reports, and once it's complete, it'll open the folder where I've sent them. And I've asked for the full set, but you can choose which ones you want. So in the case here, we have a individual PK plots, and I'm just going to show a few of these just to give you the idea, where you have the linear and semi-log view, as well as the data and any of the map data you've set. And it's, since we asked for individual plots, we have one for each, and at the bottom we have the whole data set for our view. We also generate an Excel document that has many of these reports on individual tabs, so you can copy, paste, modify it as you need to generate your own reports. So that was their simple way to make an NCA. It's just a point and click. And the new feature we added, as John had mentioned on the NCA tab, was the Non-Parametric Superposition. So when we were using this tablet data set, as we were scrolling, we noticed we have a mix of male and female. We can also subset the data and make a copy if we want, and we'll call this FASTD 140 tablet, but we'll make this just females. When we create this data set, we inherit the filters from the parent data set, but we can add one more, or as many more we want. In this case, we're going to add just a female. So now when we look at our female data set, and I'll extend this out, we have only the two that were part of that original data set. And in order to use it, we'll set our terminal slopes the same way. Let's try that again. That was none. I had the wrong data set selected, excuse me. All right, and it's the same thing. We can view them and change them as we'd like. So we're going to perform a non-parametric superposition analysis on these profiles. And in this case, we're going to do a 200 milligram loading dose. We'll do 7 doses, but we'll do an interval of 24 hours. And we'll just call this data set NPS. So what happens is the results of the non-parametric superposition, you'll see your source profile as well as the generated profile for each of the source profiles. The output is another data set. So with this data set, we can view the profiles that were generated from our NPS, and you'll see their type shows us that. And we can use these profiles and this data set to perform a NCA just as we did with our source data. So we'll look at the terminal slopes, the same as we do, and we can run an NCA. And again, you'll see our live statistics, our AUCs, everything we asked for is right there in front of us. We have the same options to select our data as well as save either our plot or our data. And moving on, we'll take a look at the multi-dose simulation. To do this multi-dose simulation, we need obviously our settings, and we also need some initial values. If you know the values you want to use, you can enter them, or you can generate them by running a compartmental analysis on the data itself. So we're going to run a 2-compartment. And we'll see our compartmental results. You have your statistics. Again, our plot is consistent. You can do all the same features you did before, save your image, select your data. You'll notice we track which points were removed by BLQ. So there you have the record of the data used, but also the BLQ treatment is live right in front of you. But we're going to use the results to run a multi-dose simulation. So what we do is we just select the compartmental analysis we want to use, select the results. Since we did a fit each, we can actually fit or choose which profile we want to use as our starting point. Then we set our simulation time, how many points per profile we would like, our data set name, and then we set up our dose regimen. So in this case, we'll do an infusion, and we'll just do 1 milligram start, interval of 24, infusion time of 5, and we'll ask for 5. Click the simulate button, and we have our doses. And again, since we have a data set as the output, we can treat it just like the source, view our terminal slopes, and run an NCA. Each of these analysis we ran are stored on our results table, and as we click, you can see the settings we use, the initial values, as well as the dose regimen we set up. You can add, remove, as you run an analysis, you can remove the ones you don't want, save the ones you do. And by picking the analysis you're interested in, you can choose which reports you want to run. We also have a custom report generator in the case of, we'll use that NCA. You have the ability, if you don't like the auto reports we generate, you have the ability to select which variables you want reported and in which order. So you can select the profile variables, or mostly the map variables, and then select the parameters you'd like to report. You can also load a saved report settings. So now in this case, we pick where we want to save it. give our report a name, and click Generate Report. And now, when you look, you'll have only the settings we asked for in the order we asked for them. So this allows you to generate standard reports you use in your analyses and save them so you don't need to constantly set up and adjust. If you have a standard template, you can run your analysis and generate your standard reports. And I think that's our quick run through, John.
Thank you so much, Mark. Very impressive. I'll take controls back here and just summarize kind of what we talked about today and then open it up for questions. So let me take back controls here. So just to summarize, PKPlus version 2 now adds command line scripting capabilities. We saw already the automated system verification runs that you can perform upon installation of PKPlus version 2. And then you can also write your own command line scripts to process in a high throughput manner the analyses for your PK profiles. And you can work with some of the items that Mark mentioned, especially towards the end. The custom report templates that you save can be utilized in these command line scripts to produce in this automated fashion a set of reports for a variety of PK curves that are loaded in. We've touched on the Windows authentication and user group options, so we can now manage access privileges to satisfy application level compliance for PKPlus. We've incorporated now prediction capabilities with PKPlus. So version 1 was focused on all things PK modeling, being able to do the non-compartmental and compartmental PK fitting with your data. Now we've extended that with version 2 to do the predictions or simulations. Both non-parametric superposition and compartmental PK simulation features are available to predict curves for different dosing regimens. And then, as mentioned, you can save now your mapping and report settings for reuse in other projects. And this should allow for much faster processing of your PK study data and better communication across groups. So essentially, you now have everything that you need for PK modeling and simulation functionality. All the critical features without needing to worry about where do I find this, how do I turn this on or off. There's a lot more that potentially could be done, but the vast majority of your PK modeling and simulation activities are going to be handled with PKPlus and the features and capabilities that Mark outlined in the demonstration. And just as a reminder how this benefits you, either in industry or academia, being able to utilize and access PKPlus at a low cost, sharing licenses, subscribing to licenses for both short or long-term periods to satisfy commercial needs, and for academia, getting access for free to PKPlus to help with your learning, your teaching of pharmacokinetics, and your analysis of PK data sets that you're generating through government or nonprofit grants. And so with that, we'll stop here and open it up for questions. And I think there have been a number of questions that are already coming in here. And this is where I'll also ask my colleagues, Kay and Vieira, to comment as well. So the first question that came in is with respect to accessing the software and whether or not consultants, independent consultants, can gain, how do they gain access to PKPlus? If we are working with PKPlus to support commercial activities, so if you're a CRO or a consultant that is providing a commercial support to a client who is in an active development phase, we would be more than willing to provide access to PKPlus through a subscription model. So you can license PKPlus for as short as 1 month, for multiple years, depending upon what your time frame is for the project itself. If you are a consultant or a CRO that is fully supporting nonprofit activities, and you agree to use the software by the terms and conditions of our nonprofit software license agreement, then we can talk about providing access in other ways as well. For another question that came in, for PO pharmacokinetics, for oral PK, is it possible to perform deconvolution of the absorption from the elimination? And for that, I'll ask one of my colleagues on the panel to address that question.
So we fit the absorption and then the elimination together if you're talking about the compartmental models. So the fitting itself will speed out the absorption constant and also the elimination constant.
Okay. Thank you, Kay. And then I'll just add that if we were also thinking of this, if the question is along the lines of doing deconvolutions of oral PK profiles, and then potentially using that for doing any sort of in vitro and vivo correlation work, that would be something that would be handled in the GastroPlus platform. It's not something that's incorporated into PKPlus. Another question that came in is with respect to the data and the results. So can the data or results be exported to R, or can information from R be imported into PKPlus?
Currently, the exports are only done to CSV or Excel books. You can format them in a way that's easier to read for R, but we have no direct export to it, no.
Thank you, Mark. Another question, can you solve for the compartmental model that simultaneously fits all selected profiles the best?
Yes, we can do that. So the as the Mark demonstrates, we fit each of the profiles, but there's also an option that you can fit all the profiles simultaneously using the same PK parameters.
Thank you, Kay. And then a follow-up. Once we do that simultaneous fit, can we use that for the basis of the multi-dose simulations?
Yes, we can.
Thank you, Kay. Another question is for the command line option. Do I have to write out the XML files from scratch, or can I use completed analyses to save a template directly from PKPlus?
Currently, we don't have the ability to save a project to a template, but you can use the ones that are verification package has all of the templates we use that can be used as a starting point, or we can walk you through creating your common template.
Thank you, Mark. Another question, can we use PKPlus to introduce the inhibition models, or for some drugs, auto inhibition as well?
Yeah, right now we didn't incorporate it in PKPlus yet, but in the future versions, we might have plans to include those type of models.
Okay, thank you, Kay. Yeah, for PKPlus, the current implementation of the software is really focused on the PK modeling and simulation aspects. So inhibition, PD, that's not something that's been incorporated yet into the platform. This is really focused on all things PK modeling and simulation. Another question, I know the answer already, but I'd still like to hear from the panel. There's interest in using bioequivalence study data, and we have two questions. First is, how does PKPlus deal with outliers?
Well, maybe I can take that one. As Mark was demonstrating, the outliers is something to the user to deal with, right? Mark was demonstrating nicely how you can review the data, whether there are any outliers and so on, to help you really visualize what is happening with your data set. And that gives you option to decide what you want to do with the outliers. He did a quick demonstration of how you would be defining the BLQs, but there is also an option to exclude the outlier, either points or the entire profile. So it does give you a very nice visualization and very quick options to decide what you want to do with the outliers.
Thank you, Vera. And then, do we use the 90% confidence interval for the difference in the means of the log transform data for different PK parameters between test and reference products?
Well, again, as you mentioned earlier, these sorts of extensions are not currently part of PKPlus, right? So right now it is more of a analysis of PK of the simple data sets, but the comparisons of bioequivalence has not been incorporated yet.
Thank you, Vera. Yeah, that would be something to explore more with the GastroPlus platform in the case of BEs, calculating bioequivalence or performing virtual bioequivalence trial simulations. Another question. In the demonstration, it seems the analysis was performed on nominal sampling times. Can the software handle actual times?
Yes, it can. The example data set I had the nominal times, but you can use the scheduled time and the nominal time if you have it available in the file. All statistics are done using the scheduled time, but the analysis are done using the actual time.
Thank you, Mark. And then, does it correct for time deviations by interpolation, extrapolation, and impute missing samples where possible?
When it's doing the analyses, it uses the time only the times provided for analysis. In other instances, and Kay can elaborate if I'm off, in the case of partial AUCs or terminal slopes, anywhere it needs to interpolate the data to perform the function, it does, but not as a rule when doing the NCA.
Okay, thank you, Mark. Can metabolite PK be simulated for multiple PO doses?
Right now, we don't connect the parent compound and the metabolites. What you can do is just when you create the file, you can tag your parent and metabolite and then put them in different groups. And then looking at the AUC, we can do the non-compartmental analysis on those data, but we don't connect them together in this version.
Thank you, Kay. And that would be something that we could do in the GastroPlus platform, is if we wanted to mechanistically track formation of metabolites from the metabolism of the parent, that would be something done in the GastroPlus platform. And this next question is also something that would be done in the GastroPlus platform. Can we use PKPlus to simulate drug-drug interactions? So that would be something that is done mechanistically in the GastroPlus program. Does PKPlus include models with enterohepatic circulation in its library?
I think the same answers to the previous question. No, these are simple non-compartmental or simple 1 to 3 compartment models for enterohepatic circulation or more additional mechanisms. You'd go into GastroPlus for that kind of analysis.
And then the same question for transit compartmental models. That would also be something that is done in the GastroPlus platform. A scientific or practical question in BA, concentrations can be measured as low as 1 nanogram per mil, but the interesting range is in general greater than 50 nanograms per mil. Less than 50 or around 1 to 10 nanograms per mil usually give another elimination phase. Do you use the 1 to 10 nanogram per mil data as the better phase when doing the fitting?
It's really depends on the user, as we showed that you have the option to set up your the filter for your data. It's really a user option. We don't provide a guideline for how you handle the data. We provide the functions for you to handle the data.
Thank you, Kay. And I believe that is the last question. Just a couple minutes before our end time here, so that's good. We're good on time. So let me then just kind of wrap up. I want to thank Mark, Vieira, and Kay for the excellent and insightful discussions today. And that's going to conclude the webinar. For more information on PKPlus, including requests for evaluations or pricing, or to register for this new no-cost academic license program, be sure to visit our website, www.simulations-plus.com. And also connect with us on one of our social media channels for the latest news and events across all divisions at Simulations Plus. And with that, I would like to very much thank you for joining us and hope you have a wonderful day. Thank you, everybody. Bye-bye.